In this module, Toxicology, this module gives drug safety professionals a working command of the toxicology that sits behind a safety file. It covers the principles of toxicology, the non-clinical safety package required under ICH M3R2, and the ICHS series, good laboratory practice, dose descriptors, and first-in-human dose setting. Learning outcomes. Explain the core principles of toxicology, including dose response, and distinguish acute from chronic, local from systemic, and reversible from irreversible toxicity. Describe the non-clinical safety package recommended by ICH M3R2 and the roles of the ICHS series guidelines and good laboratory practice. Calculate and interpret dose descriptors including NOAEL, HEAD, and MRSD, and explain how first-in-human starting doses are set. Evaluate the Mabel approach for higher-risk biological medicines and explain the lessons drawn from the TGN 1402 first-in-human incident. Toxicology, core principles for drug safety. Toxicology studies the adverse effects of chemicals on living systems. In drug development, it defines the hazard before any person is exposed. Paracelsus is credited with the principle that the dose makes the poison. Almost any substance becomes harmful at a high enough dose. Acute toxicity follows a single or short exposure. Chronic toxicity develops over repeated dosing across weeks, months, or years. Local toxicity occurs at the site of contact, such as injection site necrosis. Systemic toxicity follows absorption and distribution to distant organs. Dose response thresholds and therapeutic index. The dose-response relationship is the foundation of toxicology. As dose rises, the severity of effect and the proportion of subjects affected usually rise. A graded dose response describes increasing severity in one individual. A quantile dose response describes the proportion of a population showing a defined effect. Threshold effects require a minimum dose before harm appears, which is why a no observed adverse effect level can be identified at all. Therapeutic index compares the dose-producing toxicity with the dose-producing benefit. A narrow index leaves very little margin for error. The non-clinical package and ICH M3R2. ICH M3R2 recommends the non-clinical safety studies needed to support each stage of human clinical trials and eventual marketing authorization. Before first human exposure, you generally need safety pharmacology, genotoxicity, and repeat dose toxicity studies in one rodent and one non-rodent species. The core safety pharmacology battery covers cardiovascular, central nervous and respiratory function, and should generally be completed before human exposure. Repeat dose study duration broadly matches planned clinical duration, up to 6 months in rodents and 9 months in non-rodents for long-term use. The ICH safety guidelines at a glance. ICHS1 addresses carcinogenicity testing, S2R1 genotoxicity, S5R3 reproductive and developmental toxicity, and S6R1 biotechnology-derived pharmaceuticals. S2R1 offers 2 equally acceptable batteries. Option 1 adds an in vitro mammalian assay. Option 2 assesses genotoxicity in vivo in 2 tissues. Both S2R1 options begin with a test for gene mutation in bacteria, commonly known as the Ames test, as the first screen. S5R3 sets the strategy for detecting effects on fertility, embryo-fetal development, and prenatal and postnatal development, including male fertility. GLP and the OECD principles. Good laboratory practice is a quality system covering how non-clinical safety studies are planned, performed, monitored, recorded, reported, and archived. The OECD principles on good laboratory practice, number 1 in the OECD GLP series, are the internationally recognized standard for regulatory safety studies. GLP is about data integrity and reconstructability. It proves the study happened as reported. It does not by itself guarantee good scientific design. Each study has a named study director with overall responsibility, supported by an independent quality assurance program that inspects and audits. Dose descriptors: NOAEL, LOL, MTD, HEAD, MHSD. NOAEL is the highest dose tested in an animal species that produces no significant increase in adverse effects compared with the control group. LOAEL is the lowest dose at which an adverse effect is observed. It is used where no clean NOAEL could be established. Maximum tolerated dose is the highest dose animals tolerate without unacceptable toxicity, used mainly in oncology and in carcinogenicity study design. Human equivalent dose converts an animal dose into the equivalent human dose using body surface area scaling rather than simple body weight scaling. Worked example: NOAEL to HEAD to MHSD. FDA guidance converts an animal dose to a human equivalent dose by dividing the animal dose in mg per kilogals by a species division factor. The published division factors are 12.3 for the mouse, 6.2 for the rat, 3.1 for the rabbit and the monkey, and 1.8 for the dog. Illustrative case: a rat NOAEL of 50 min per kg per day gives a head of 50 divided by 6.2, which is 8.06 min per kg. A dog NOAEL of 15 min per kg per day gives 15 divided by 1/8, which is 8.3 mol per kg. The rat is therefore the more sensitive species. First-in-human dosing and the Mabel approach. The EMA Guideline on Identifying and Mitigating Risks in First-in-Human and Early Clinical Trials, Revision 1, was adopted in July 2017 and applied from February 2018. Mabel is the minimal anticipated biological effect level, derived from pharmacodynamic data including ex vivo and in vitro studies in human tissues where feasible. Where uncertainty about the human relevance of animal findings is high, the guideline expects the starting dose to relate to Mabel rather than NOAEL alone. The starting dose in healthy volunteers should give exposure below the pharmacologically active dose, unless a robust scientific rationale supports going higher. TGN-14.2: What happened and what changed? TGN-14.2 was an anti-CD28 monoclonal antibody administered to 6 healthy volunteers at Northwick Park in London in March 2006. All 6 developed a severe cytokine release reaction within hours, progressing to multi-organ distress and requiring intensive care support. The non-clinical package had not predicted the reaction, so the case remains the standard illustration of failed species translation for an agonist biological. The primary published account is the case report by Suntharalingam and colleagues in the New England Journal of Medicine in 2006. The 3 Rs and new approach methodologies. The 3 Rs are replacement, reduction, and refinement, first defined by Russell and Birch in their book The Principles of Humane Experimental Technique. The NC3Rs is the United Kingdom National Organisation for the 3 Rs, funding research and producing training and resources to advance them. Replacement means using predictive models and tools instead of animals. Reduction means designing robust experiments so fewer animals answer the question. Refinement means minimizing pain, suffering, and distress, and recognizes that animal welfare directly affects the quality of the scientific result. Drug-induced liver injury and Hy's law. Drug-induced liver injury is a leading cause of drug withdrawal and a leading cause of acute liver failure in Western countries. The FDA guidance on drug-induced liver injury, issued in July 2009, sets out how Hy's law is applied in pre-marketing clinical evaluation. A Hy's law case requires hepatocellular injury, generally shown by ALT or AST, at least 3 times the upper limit of normal. It also requires total serum bilirubin above twice the upper limit of normal without initial findings of cholestasis judged from alkaline phosphatase. Worked example: an illustrative Hy's law check. Illustrative figures only. Assume laboratory upper limits of normal of ALT 40 units per litre, Total bilirubin 20 micromole per litre, ALP 130 units per litre. Day 28 results show ALT at 260 units per litre, which is 6.5 times the upper limit of normal, so the hepatocellular criterion is met. Total bilirubin at 55 micromole per litre is 2.75 times the upper limit of normal, so the bilirubin criterion is also met. ALP at 150 units per litre is 1.15 times the upper limit, well below twice, so the pattern is hepatocellular rather than cholestatic. Kidney, heart, blood, and immune toxicity. The kidney is vulnerable because it receives high blood flow and concentrates solutes in the tubules. Aminoglycosides and cisplatin are classic nephrotoxins. Cardiotoxicity includes direct contractile injury, as seen with anthracyclines, and electrophysiological effects, such as prolongation of the QT interval. QT prolongation can precipitate torsades de pointe, a polymorphic ventricular tachycardia that may deteriorate into ventricular fibrillation and sudden death. ICH E14 governs the clinical evaluation of QT prolongation and ICH S7B the non-clinical. The 2022 E14 S7B questions and answers describe a double negative strategy. From animal findings to human risk and the RMP, non-clinical findings identify hazard. Whether a hazard becomes a human risk depends on exposure margin, species relevance, and the underlying mechanism. Translation fails in both directions. Some animal findings never appear in humans, and some human reactions were never seen in any animal species. Immune-mediated type B reactions, idiosyncratic liver injury, and rare hypersensitivity are poorly predicted by conventional animal toxicology programs. The Risk Management Plan safety specification includes a non-clinical part, covering findings not adequately addressed by the clinical data. That completes this module. The key takeaways and suggested further reading are summarized on the slide. The full references for this module are listed on the slide for your further reading.